Welcome, everyone. This seminar consists in 2 research talks. Each speaker will deliver a 30-minute presentation with an additional of 5 minutes for questions, and there will be 20 minutes in the end for a final discussion. We hope you have the opportunity to interact with our guest speakers and ask them lots of questions. This seminar is about blood-feeding arthropods. Today, our guest speakers are Dr. Pablo Schilman from the University of Buenos Aires and Dr. Maarten Voordouw from the University of Saskatchewan. I don't know if the pronunciation of his name is correct or not.
That was fine. Thank you.
Okay, thanks.
Hello.
Hi,
Hello.
Hello.
Well, Dr. Schilman's presentation is titled Thermotolerance in Vectors of Chagas Disease. Dr. Schilman is a CONICET researcher and a professor at the University of Buenos Aires. His main area of research is insect eco-physiology. He's interested in the physiological adaptation of insects to the environment, and in particular, how abiotic factors such as water availability and temperature affect the behavior and physiology of insects, and which are their physiological limitations for the geographical distribution. And Dr. Voordouw's talk is titled The Ecology of Pigs that Transmit Lyme Disease, a Tale of Mice and Time Lags. He has been an assistant professor in the Department of Veterinary Microbiology at the University of Saskatchewan since 2018. His work focuses on host-parasite interactions, vector-borne disease, multiple strains pathogens, and the ecology of mixing infections, Lyme disease and other tick-borne infections. So now I'm going to stop to share my presentation, and Pablo, you can start to share yours.
Thank you, Vanessa. It's okay. Everybody's watching the presentation.
I can see it.
Okay. So maybe I don't know. Maybe I will stop the video, or do you prefer that I keep the video or stop it? Because I can keep it, and in case the connection is not so good, I will stop it.
Okay, that's totally up to you.
Okay, thank you. So, well, I will talk about thermotolerance in. We put the time because I'm not very good at time. So, the thermotolerance in vector of Chagas disease. Okay. These 2 hands represent, for one hand is the ecology, the other is the physiology, and one drew the other one. So the ecology will concentrate, or will focus in patterns or process and patterns of distribution of species. And on the other hand, the physiology will try to make to understand the interaction between the animals, and also the mechanisms that can underline these patterns that we observe. So basically, the framework of this study is the macrophysiology. That is like a new field of research. It's almost like 20 years since it started, and it's the combination of 2 other research fields of research that like ecophysiology and macroecology. So based on the definition by Stephen Chown, the macrophysiology is the investigation of variation in physiological traits over large geographical and temporal scales and the ecological implication of this variation. So in general, we can say that animals and plants are limited in their distribution by their physiological adaptation to biotic and abiotic factors. So, and these abiotic factors are mainly the humidity and the temperature. In particular, in my talk, I will focus on temperature, and so the effect of temperature and the tolerance to temperature, how this will affect the distribution of insects, among other things. Also, the temperature changes. So there are like changes in space and in time. So in space, for example, there is a temperature gradient with latitude and altitude. So higher latitudes or altitudes will have lower temperatures, and there are also some changes in temperatures during the year, like the seasonal changes, and in shortest periods of time, for example, for the daily temperature variations. So these temperature variations, the temperature itself will affect a lot to the animals, in particular to ectotherms, that the body temperature match the environmental temperatures. And so they will affect many, many different processes. And insects are, well, not all, but most of the insects are ectothermic animals, and so they will be very affected by these changes in temperature. A couple of things that I want to mention that maybe everybody knows, but I just learned a few years ago, are these two things. One is the Rappaport rule. The Rappaport rule, it was named because of Eduardo Rappaport, who was an Argentine ecologist, and proposed that there is a positive correlation between the size of the organism's geographic range and its latitude. And this rule was tested in many different taxa, and most or many studies support these rules while others doesn't. So, and this plot here is from an old study with birds, and you can see here on the x-axis the latitude, and on the y-axis the area of geographical range, and you can see that there is a positive correlation between the two. Sorry. On the other hand, the climatic variability hypothesis can be used to explain this Rappaport rule. And why is that? Because the climatic variability hypothesis states that terrestrial organisms are distributed in highly variable environments, for example, high latitudes and or altitudes, are adapted to withstand a broader range of climatic conditions than organisms living in less variable environments. That's why the organisms that is living in higher latitudes, for example, will have a wider or a bigger change, for example, in temperature during the year or during the day. And this capacity of the organisms to live in that situation will allow that they have a bigger area of distribution. Other thing that is important to mention before I start with the results is this curve. This is a performance curve. The performance curves are used for many processes, so you can plot any biological or any process here and how it changes as a function of temperature. In this case, it's the activity. And so we can see that at the beginning with low temperatures, there are no activity at all. And then at a certain point, the activity starts. This point is called the critical thermal minimum, or CT min. And then with increasing temperature, there is an increase in activity up to a point where the activity is maximum. And this point is at the optimal temperature. And then if the temperature continues to increase, there is a sharp drop in the activity. until it stops. At this point is the critical thermal maximum. These 2 points, the CT max and the CT min, will, so the difference between these 2 points will give us the temperature tolerance range. So that's the temperature where, in this case, an insect will be active or an animal will be active. And that's why these measurements are useful for some ecological studies. I will present data with these triatomines that are vectors of Chagas disease. There are more than 140 different species living all in America, mostly in the intertropical region. However, there are some species that lives in more temperate regions with cold winters, like in Patagonia or in the US, in Indiana and Maryland. It's not so cold as they are in Canada, but it's colder than the tropics. And these insects are hemimetabolous insects. They have, here you can see one of the species, Rhodnys prolixus, it has the egg, and then 5 different instar stages, and finally the adult stage. And I will present data all with this last instar stage, that is the 5th stage. So the effect of temperature or heat are known in many or several species of triatomines in different behavioral and physiological processes, like the thermopreference, or as a clue to find food or find a host, also in reproduction, also in the rate of development or metabolic, or also metabolic rate, learning and memory, and also as a climatic variable in the geographical distribution. However, until we start with these studies, there were no information on thermotolerance in any vector of species of Chagas disease. So the aim of this first part of the talk will be to explore the possible relationship between thermotolerance and the geographic distribution of seven species of Chagas disease vectors that are epidemiologically important and with very different latitudinal distribution, to test the climatic variability hypothesis that explains the Rappaport rule. So for doing that, we combine 2 different methodological approaches. One is the species distribution models, and the other is to measure, analyze, and compare individual physiological tolerance, in this case, thermotolerance. So first, we model the ecological niche of each species using the bioclimatic variables that we found on internet, for example, in WorldClim. And then we identify the most important of these abiotic factors for their distribution. Then we measure and analyze and compare the thermotolerance for the 7 species, measuring CT max, CT min, the upper lethal temperature, and chill coma recovery time. And finally, in order to unravel possible causes for the distribution of Chagas disease vector, we analyze whether there is any link between the most important abiotic limiting factors and the thermal tolerance ranges for the species to test the climatic variability hypothesis. So as I said before, we study 7 different species that were chosen because of the different distribution, also because their phylogenetic relationship, and more important, because of the epidemiological relevance. For example, Triatoma infestan is the most important vector of Chagas disease in Argentina and also in South America, like Bolivia. And Rhodnys prolixus is the most important vector in the north of South America, and is also a model a model of study for insect physiology. So here, first we perform this habitat suitability maps with Maxent. So Maxent is an open software that makes these correlative models between the bioclimatic variables and the only presence of the insects. So we have these 7 mass for each species, and with darker brown being the more suitable habitat for the species, and when goes lighter with less suitability. And then with the same software, with Maxen, we perform, or we analyze which are the limiting, the limiting abiotic factors, and we can see, I cannot see the, here for at least these 4 species, and maybe Triatoma sordida too, Rhodnius prolixus, Triatoma dimidiata, infestans, and Patagonica, that the bio-6 is a very important limiting factor, and bio-6 is the coldest, the coldest day of the colder, is the minimum temperature of the coldest month, sorry. So then we make the physiological measurements. This is the chill coma recovery time. We place the insect 4 hours RCK at 0 degrees Celsius, and then we put it at 28 degrees upside down and measure the time until the insects turn to the normal position. And what we can see is that species, these 3, that they didn't recover in 1 hour, 28. No one was dead, so they at 24 hours later, all the insects were alive. But it was not enough for 1 hour, and these are tropical species. And on the other hand, these 3 recover in about 10 minutes, and this Triatoma patagonica, that is the species that has the most southern distribution, reach Patagonia, like his name, it's his name, recover in about 5 minutes. Then we measure the critical thermal minimum, or CT mean. For that, we perform this ramp of decreasing temperature at a rate of 0.25 degrees Celsius per minute, or minus 0.25. In this plate that is with a temperature controller, and we market the insect did not respond to a mechanical perturbation. And these are the data for the 7 species. We can see that Triatoma infestans and Patagonica are the 2 more tolerant to cold, again. And others like Rhodnius prolixus is not so. So there are almost like 6 degrees difference between these 2 species. Then we measure CT max, or critical thermal maxima, with a different methodology. This is by open flow respirometry. I will not get into detail of this, but basically we can measure individual insects in real time. We can measure activity, CO2 production, and also water release and the temperature in real time for individual insects. And we perform an a ramp increasing temperature of 0.25 degrees per minute. And we have this kind of recordings where you can see in red the temperature ramp with increasing temperature. Then in black trace, we have the CO2 release or production of the insect. And then in green, we have the activity. And these 2 are just the analysis of the CO2 and the activity. That is the absolute differential summation. That is, for this, for example, we do all these signal positive and then make this the accumulative sum of this. So that's mean that there is an increase of the slope of increasing activity will show that there is an activity, and when there is no activity, it will be a flat line. With this, we calculate the CT max, where there is not such a clear pattern for the species. For example, Triatoma infestans and Triatoma patagonica are quite in the middle, and there were the two that has more, that they were more cold tolerance. So when we make the temperature tolerance range, that is the CT max in red minus the CT mean in blue, we see the degrees, the temperature range that the insects could be active, and is maximum for Triatoma infestans, Patagonica, and Sordida, with about 46 or 47 degrees of range. And then, in order to find a link between the abiotic factors and the physiological traits, we perform this. We plot we perform these plots. There are 7 plots, one for each species. Here is the minimum temperature of the coldest mouth, so that means the bio 6. And here is the maximum temperature of the hotter mouth, and this is the bio 5, I think. And so the points are the distribution of the insect, and the colors are the suitability by Maxent. So you have red color is maximal suitability, yellow is not so good, and white is not good. And the ellipse enclose the 95% of the data. And then we have in red dashed line the CT max of this species, and in vertical blue dashed line the CT mean. And we can observe first that for the 7 species, the distribution, the locations, the distribution, the places where the insects inhabit, has the maximum temperature of the hottest mouth is always lower than the CT max. On the other hand, the minimum temperature of the colder mass is not always lower than the CT mean. For example, in Triatoma delponte and Triatoma infestans, there are places where the lowest temperature of the coldest mouth is lower than the CT mean. And so when we put all together in these maps as a function of latitude, we can see that the maximum temperature of the warmest mouth is more or less constant. There is no big changes. The same for the CT max for the species, while there is a negative correlation that is obvious of the minimum temperature of the coldest month with latitude. So increasing latitude, more close to the poles, will be colder winters, and the same a little bit with CT means, and some species that lives at higher latitudes will have will inhabit some places where the coldest temperatures are below the CT mean. So to summarize this first part, we can say that these were the first measurements of thermotolerance in vectors of Chagas disease, that the microphysiological approach used agrees with the climatic variability hypothesis, and that there was a greater thermal tolerance range for species at higher latitudes, and subtropical species can reach some areas with temperatures lower than their CT mean, while tropical species would be limited by their distribution to areas where the minimum temperatures are higher than their CT mean. So the distribution of insects depends, as I said before, on biotic and abiotic factors, and among which tolerance to cold is an important factor. In this case, combining species distribution models with physiological tolerance or some mechanistic approaches allows to understand the ecological niche of the species, and that will allow to predict where the limiting temperatures will occur in the future, because, I mean, with some. The thing is that if you have a species that is an invasive species that will change from one place to another, or in the same place with global climate, global warming, the temperatures will change, and so knowing more the physiological limitation will allow us to understand better or to predict better if what will happen with the distribution of a certain species. So we show that for triatomines, cold tolerance is more important than heat tolerance in determining their geographic distribution. However, the upper lethal temperature is very, very close to the city max. And you can see this in this plot where you have the 7 species, and in red we have the city max for the species, and in black you have the upper lethal temperature. And you can see that in most cases there are no significant differences. There are only differences in these 2 species, in Triatomys and Rhodoplex, but the difference is about 1 degree. It's very small. So that's why it's very important that the insects were well below the city max, because it's very close to the lethal temperatures. And so how can the insects do that? There are many mechanisms of body temperature regulation in insects. For example, some insects produce heat. There are some dung beetles that produce heat in the thorax, and they can be active at very cold weather or at low temperatures. Also, some bumblebees can fly in some at temperatures that it will be physically impossible, but they warm up the fly muscles in order to be more optimal for their activity, and they can fly. Also, there are some behavioral adaptations. So the insect will move from one microsite to another in order to find a certain microclimatic condition that is more favorable, that is better than the other one. And also there are some mechanisms of evaporative cooling. That is there are, for example, some cicadas that sings in the worst, also in the hottest hours of the day in the desert, and by evaporative cooling they can continue to be active at that temperature. Also, there are some mosquitoes when they feed blood that is warmer than the environment. They release a drop of urine that will evaporate and will cool down the mosquito body. And what about triatomines? So it's known that when triatomines ingest blood, also there is an increase in the expression of heat-shock proteins. And also it seems that there is some kind of countercurrent system in order to cool down the temperature of the blood when gets into the insect. But what about the evaporative cooling? Here we have some pictures that were taken under a microscope. Here you see is one Rhodoplex prolixus. There are these two, the 2 compound eyes. Here is the proboscis. Here are the legs. And at the tip of the proboscis, I have my picture here, I cannot see. If you see, there is some kind of liquid. This is some saliva. This appears when you heat up the plate where the insect is on. And so the aim of the next part of the talk will be to show that the mechanism of evaporative cooling can extend the upper limits of thermal tolerance in Rhodoplex prolixus, one of these 7 species. For that, we did these experiments where we closed the proboscis with an epoxy putty or putty, I don't know how to pronounce. And then we have some control that we have the same epoxy, but on one leg. And we measure by open flow respirometry the CT max. And also we measure, in this case, the release of water. So here you can see this is the time. This red line is the increasing in temperature. In blue, we have water, and there is one peak of temperature here. And in green, we have the absolute differential sum of the activity. So at this point is where from being active start to be inactive. So at this point will be the CT max, that in this example is 43.4 degrees Celsius. And for treated insects, we can see two things. First, that the CT max is more than 1 degree below the other one. And another thing that is important is that the peak of water is smaller than this one. And we can quantify this in many different recordings by doing the differential of this curve. So we can calculate the area under the curve for this kind of recordings. And doing that, we found that the treated insect release less water in this peak than a control one. And this is reflected in a change of the CT max. So being 1 degree significantly lower for the insect that was covered with this epoxy in the proboscis. In addition, some infrared video camera will show here is one insect that is on a surface that is getting hot. Here, I don't know if you can see, there are the two antenna, the head, and here is more or less the tip of the proboscis. Here are the legs. So it will color because it's increasing the temperature, and there is a delay in the temperature of the insect compared with the background. And when it starts to get it hotter, it will start to release saliva here and will be cool. There it is, there, again and again. And if we use just one frame of this video and make one line from point A to point B and calculate the temperature along this line, we can see here is point A. We can see that it's almost 46 degrees. And then when it reaches this point where the tip of the proboscis is releasing the saliva, there is a drop in the temperature of more than 3 degrees. And so this release is not in any part, so it's not that you are cooling down a very small spot, but it's not in like in the abdomen or in a leg, but it's in this place that is where the central nervous system is. So it's an important and essential part of the insect. So this change in 1 degree in the CT max could seem, so it seems small, 1 degree, but 1 degree is enough for the insect to have more time to escape from this place and to be alive. And so that's, I think, is an important point. So to summarize these things, we can say that the mechanism of evaporative cooling would be used by Rhodochrolixus, allowing it to extend its thermal tolerance range and thus be able to escape lethal temperatures. The macrophysiology approach make it possible to study some mechanisms by which large-scale patterns of species distribution are generated, for example, to test the general ecological hypothesis or to predict the geographical distribution and spread of invasive organisms or the transmission of diseases as a result of global climate change. And I would like to thank Berenice and Vanessa for inviting me here. Also, my laboratory, the Laboratory of Ecophysiology of Insects and the Laboratory of Insect Physiology, that we made some things together. The la agencia, la Universidad de Buenos Aires and CONICET for some financial support along the years. And the results were performed basically by Gerardo de la Vega, who was a PhD student in the lab, and then by Agustín Álvarez Costa, who is actually a postdoc in the lab, and Jose Crespo, who made the infrared video. If you have any question, I'm happy to answer if I can.
Okay, so everyone, now it's time for questions. If you have a question, feel free to use the chat box or unmute yourself and ask whatever you're thinking about.
This one in the chat? Or?
Yes, it says, would any of these species experience sub-zero temperatures, perhaps in alpine environments? And if so, have you considered measuring the supercooling point, the temperature at which the insect freezes?
Yeah. So, yes, I imagine there are, for example, Triatoma patagonica are in places where there are these kind of temperatures. And maybe it will be, yeah, it will be nice to measure in supercooling points. However, some of these, it's also important, I mean, to have the measurements of temperature in these micro habitat places. So it is, for example, some species that are more epidemiological, more important are because they are associated with humans, so they live in the house of the people, so that's why they can transmit disease more easily. And so it's not always that the temperature in the macro temperature will be exactly the same. But yes, I'm sure that there are these species like Triatoma. It's not, I mean, it's not that the more common things, but for some species like Triatoma patagonica, I'm sure that it will suffer for these temperatures, and it will be nice to measure this. Thank
you. Do we have any other question?
Yeah, I have a question. A really interesting talk. I was wondering in the beginning, when you're doing the Maxent modeling of the species range, and I was wondering, I mean, was that mostly just based on temperature, or are there other abiotic variables that were also considered, like is relative humidity important to this species and so on?
Yeah, so basically we start using the whole variables, and then we just takes bio 6 and bio 5, that is the maximum temperature of the hotter month and the minimum temperature of the coldest month, and make correlations, and all the variables that were in close correlation with that, we discarded, and we left the others that were not so correlated with this one. And yes.
Okay, so mostly based on the temperatures then. But do you think that other abiotic variables? I mean, what would you say besides that? Are there other important variables? Like, are they host specific? These triatomine bugs, and do they have, or are they generalists and they'll happily feed on anything?
There are some species that are more specialist, but they will be generalists. But there are some species that are a little more specialist. In relation to the other things, we also performed some experiments with the tolerance to desiccation, and this made this kind of it's not the same, but something similar with desiccation. And so we measure like the amount of water that the insect has, how much can they lose until they be moribund, and what is the rate of this water loss. And we calculate that these are these insects are really very resistant for desiccation. So except for one species, the other resist a lot of time. So it's not a limiting. It's not. I think it's not so limiting for these insects. Of course, the water is important in other species. So I used to work with the Argentine ant, Linepithema humile, and in that case, the water availability is very important in the distribution, and these are insects that are very leaky, so they lose a lot of water.
Right.
The same with Drosophila, for example, but not with these ones in particular. So yeah. You have to see case by case. Yeah.
All right, thank you.
You're welcome.
Do we have any other questions?
I have one.
Okay, go for it.
Thanks, Pablo, for your talk. I was wondering if did you think with Austin if it's possible to make a behavioral experiment where you make impossible for the blood-sucking insect to take out this saliva and to see what happens with the behavior if they can't lower their temperature.
Oh, that's basically more or less what I show at the end. That was we closed.
But with the animal free,
Well, the animal was free actually. It was a different experiment. It was not the one with the infrared camera, but the other was on the. Well, it's free inside a chamber, but they can move. Maybe it's a little. They can move a little less because of this epoxy, but it was the same for the control. That's why we put the epoxy on the leg. But yes, they are free basically. They can move.
All right. I was thinking more like if they are not tied in any way.
Yes, well, they were tied with the infrared camera. With the other experiments, they were in a small chamber, but they can move. So, yeah.
All right. Thank you very much.
You're welcome.
Okay, and if there are any further questions for Pablo, we'll discuss them in the last 20 minutes. Now we'll move on to Maarten. Thank you, Pablo.
You're welcome.
Okay, so I'll share my screen here. Let's see. So can people see that?
Yes.
Okay, great. So, well, thank you. I'll start with thanking the organizers, Vanessa and Berenice, for inviting me to give a talk. So I'm also going to thank Pablo because he gave a very nice general introduction to the ecology of arthropods that will help my talk. So I'm going to be talking about a different arthropod vector. I'm going to be talking about ticks, and I'm also going to talk about masked mice and time lags. I'll explain in a little bit what I mean by that. So I'll jump right into the disease here. Lyme disease is a tick-borne disease, and we often write in our introductions that it's the most common vector-borne disease in the Northern Hemisphere. You can see that it occurs in North America. It's also common in Western Europe and Eurasia. And in case you're wondering what this circle here is in the Southern Hemisphere, there's actually a Lyme disease system that's maintained in seabird populations. So things like albatrosses and shearwaters and petrels, they are infected with this Lyme disease pathogen, and there's a seabird tick that maintains the pathogen in those bird populations. But generally, from a human public health perspective, it's more of a concern in the northern hemisphere. And there's currently concern about Lyme disease and tick-borne diseases in North America and Europe because the incidence of human cases is increasing, as you see here in this slide from the CDC, where we have data from 1996 to 2016, and you can see that the cases are doubling over this time period. There's been a similar change in Canada, a very, very dramatic change in the increase of Lyme disease. And I should point out that the data shown here is probably is believed to be a big underestimate. So we think that the actual caseload is about 10 times higher. So the CDC thinks there might be as many as 300,000 people a year in the United States that have Lyme disease rather than 30,000. So because I think some people are from Canada, I thought I'd just quickly put the story in a Canadian context. So in the early 90s, Lyme disease was very uncommon in Canada. We only knew of one population of the tick vector, the black-legged tick that transmits it. That was located somewhere here in southern Ontario. So just one known tick population at the time. And now we can find this tick species everywhere or all over southern Canada, especially in southern Ontario, southern Quebec, the Maritimes, and Manitoba. So the incursion of the black-legged tick into the southern parts of Canada is one of the better examples that I know of how climate change is changing the distribution of an arthropod vector with important consequences for human public health. But this phenomenon is also occurring in Scandinavia, where the tick species that transmits Lyme disease over there is also increasing its range. I'll just say a few things. I know this is an arthropod-minded audience, but I'll just say a few things about the disease itself. So it's a multi-system disease. One of the most diagnostic symptoms is this, what we call the bullseye rash or erythema migrans, that occurs where the tick has bitten the person. And you see this ring, it actually spreads, becomes bigger over time. They think that's the bacteria spreading away from the tick bite site. And that is one of the most diagnostic symptoms. It occurs in the first few weeks following the tick bite, and about 70% of people who go on to develop Lyme disease have it. So you don't always see it. Fortunately, Lyme disease is quite easy to treat with antibiotics, but if you don't catch it, it can cause a lot of problems, and it's a chronic disease, so you'll never get rid of it. You have to take antibiotics to get rid of it. Your immune system will not clear it by itself. And so if you don't get the antibiotics, then one very common symptom is arthritis and joint pain, because the bacteria like to hang out in the joints and the synovial fluid. There can also be nervous system involvement. So sometimes people develop what's called Bell's palsy, where half of their face is paralyzed. And in Europe, there's one species that causes problems with the skin. So old people get thin, papery skin. So it's not a disease that kills you, but it is a disease that can really make your life very miserable if you leave it untreated, and will, really drastically reduce your quality of life. And this is the causative agent. So it's a bacterium. It's a spirochete bacterium. It was discovered in 1982 by a Swiss researcher named Willy Bergdorfer. So the bacterium was named after him. He was working in the United States at the time at the Rocky Mountain Laboratories. And since its discovery in 1982, we've realized that there's many different species of these bacteria. But they're all transmitted by the same group of ticks, and not all of these bacteria cause Lyme disease in people. Some of them just, are in closed cycles with certain vertebrate reservoir hosts. So We'll talk more about that in the next slide. Because, when you're talking about a vector-borne disease, you always have three players. You have the pathogen that causes the disease. You have the arthropod vector that transmits the pathogen, which in this case are ticks that belong to the genus Ixodes. And in North America, we have two tick species, the eastern black-legged tick and the western black-legged tick, which are found on different sides of the Rocky Mountains. And then in Europe, we have a tick, which is called the sheep tick or the castor bean tick. The Latin name is Ixodes ricinus. That's actually going to be the main protagonist of today's story. And that's the tick that's responsible for Lyme disease in Europe. And then what's also important to know is that Lyme disease is really a zoonotic disease. So that means that it's a disease of wildlife. And the main vertebrate hosts that this bacteria likes to hang out in are small mammals like rodents and also birds. Okay, so in nature, Lyme disease is maintained between these ticks and these vertebrate reservoir hosts. And humans and dogs, which also get Lyme disease, are really accidental hosts. And maybe one reason why people and dogs get so sick is because they're not the intended host of this disease. Okay. So, and here, this is the life cycle of Lyme disease. I'm going to spend a little bit of time here on this life cycle because it's also the life cycle of ticks. Because it's important to understand the ecology that I'm going to be talking about, and especially the time lags. So the first thing I'll point out is that the life cycle of these tick species is quite slow. It can take 2 to 6 years to complete, with long periods in between stages. And that's why, as you'll see, these time lags become important. So I'm going to start here with an adult female that's taken a blood meal from a deer, and she lays eggs. And it doesn't matter if she's infected. It doesn't matter. She won't transmit those spirochetes to the eggs. Okay, there's no vertical transmission. Some pathogens have this, but not Lyme disease. These eggs will hatch into larvae that are uninfected, and they'll become active in the summer. And if they happen to feed on a rodent host that's infected with the spirochete, shown here by this squiggly line, then they will develop into the next stage, which is called a nymph. Okay, and you see here that this nymph is also infected. And this nymph will generally overwinter. It won't try and get a blood meal that same summer. It'll overwinter and it'll become active the following spring. And if it then happens to feed on an uninfected mouse, it can transmit that infection. So the uninfected mouse now becomes infected. And that blood meal will allow it to molt into an adult tick. And again, that adult tick can overwinter and search for a blood meal the following year. So an important point here is that the larval blood, so first of all, sorry, I should say it's the larvae and the nymphs that are important for maintaining Lyme disease in nature because they feed on the same hosts. Adult ticks feed on deer, which are actually incompetent for spirochetes. They kill the spirochetes. So the adult ticks are not important for maintaining Lyme disease, other than the fact that they make baby ticks. So it's these two immature stages, the larvae and the nymphs, that maintain Lyme disease in nature. And what's important to remember is that the larval blood meal and nymphal blood meal are usually separated by about a year, right? So we generally don't measure the larvae because they're hard to find. They're very small. But what's important to understand is the conditions and the food supply for larvae, so the rodent density that determine larval feeding success, we will see that the following year when the nymphs become active, right? So if larvae do very, very well and have high feeding success, then a year later, we'll see lots of nymphs. Okay, so just a few more words about the ecological drivers of tick populations. So as Dr. Schilman alluded to, abiotic factors are very important for arthropods. We also have, of course, biotic factors, which are going to include the vertebrate host community. So this shows the importance of abiotic factors. So here we've got the developmental rate of different stages versus temperature. And so we can see that as the temperature goes up, the developmental rate speeds up. So these these tick stages will molt quicker from one stage to the other. And this is really important for determining their population abundance. And so temperature is important. In contrast to the triatomine bugs, relative humidity is very important for ticks. So this particular group of ticks is very sensitive to desiccation. It's not true for all ticks. There's some ticks that can live in very dry places, but these ticks really like to live in deciduous forests where there's a nice big leaf litter that is very humid, and they use that to meet their water requirements. And so here, this is a geographic distribution of the tick species that we're going to be talking about today. So this is the Ixodes rucinus, the tick species in Europe. And you can see that, it's found pretty much everywhere in Western Europe, not in the very far north, colder places, and also not in these, drier places. So both temperature and relative humidity and obviously other factors are important for determining its geographic range. So those are the abiotic factors, but biotic factors are also important, right? Everything needs food, including ticks. And so for ticks, they feed on vertebrate hosts. They take blood meals. They get all their nutrition from those blood meals. And so this shows the relationship between the density of chipmunks, which are an important host for ticks in North America, and the density of nymphs. And again, what's important to understand here is this time lag, right? So this is the chipmunk density in year T-1, and then the following year, we can measure the density of the nymphs. So those nymphs would have been larvae the preceding year. They got their blood meal as larvae the previous year on the chipmunks, and then we see them the following year as nymphs. And this is from a study in North America by Rick Osfeld, who's very well known for, he's an ecologist, and he's very well known for all of his work on ticks and tick-borne diseases in North America. So, but of course, for those of you that might be familiar with rodents, you know that rodent population size is not constant over time. They can be highly variable, and part of the reason why rodent populations are variable are also because of variation in their food supply. And so many, small mammals rely on the production of seeds or fruits by deciduous trees. So I'm talking about acorns that are produced by oak trees, or beech nuts that are produced by beech trees. Now, what's important to understand about this food source is that it's highly variable in time. And I'll point out here that this is where the word mast is an Old English word that refers to the fruit of these deciduous trees. So it can be used to describe acorns or beech nuts or whatever. And the next slide, I show how variable the production of this fruit by deciduous trees can be. So this is a 40-year time series on the European beech from England. And you see the time here on the x-axis, and on the y-axis, they have the amount of beech nuts produced by these beech trees. And you can see that there's this periodicity of about 3 years, where every 3-4 years or so, you have this massive synchronized production of beech nuts, and then it crashes and is quite low in the year in the time afterwards. And so what's interesting is that these trees have synchronized reproduction, even though they're of often very different ages, obviously. And that synchronization is due to weather factors, climate factors. And so, as I said, every couple of years, they'll have this massive masting event where they'll produce a lot of fruit. And so it was actually Rick Osfeld in North America who came up with this idea that, given that these acorns or beech nuts are very important for rodent populations, we can imagine that in the year of a masting event, the mouse population is going to do well. And because of the way mouse population dynamics work, you'll actually see most of the increase the year after. Okay, so in the year after a masting event, you'll have lots of mice. And then the year after that, you should see lots of nymphs, because these nymphs will have had a great time feeding on all these mice in the previous year. So we're expecting a 2-year lag time between masting events and nymphs. And Rick Ostfeld was the first to present and test this idea in North America. And interestingly, there's the North American Lyme community and the European Lyme community don't always see eye to eye on everything. So this idea was first put forth in 2006 by Rick Ostfeld, and then it's now people are interested in testing it in Europe and seeing whether that's the case there. So that's what I'm going to talk about today. Because before I came to the University of Saskatchewan, I was an assistant professor at the University of Neuchâtel, which is located in Western Switzerland, a nice part of the world, and surrounded by deciduous forests that are very good habitat for the ticks that transmit Lyme disease. So Switzerland has quite a high incidence of Lyme disease. They have about 12,000 cases a year, and their population is only 7 million. By comparison, in Canada, we have about 1,000 cases a year, and our population is 5 times bigger. So lots of ticks in Switzerland, lots of Lyme disease. And I want to start off by acknowledging my collaborators. So Lise Guerin was a director of research at the University of Neuchâtel, and she spent her career working on ticks and tick-borne diseases and made many important contributions to the study of Lyme disease. And it was really she had the foresight to start this long-term monitoring study for more than 15 years. And by the time she retired, she was, wanted to enjoy her retirement, and she basically just gave the data set to me and said, go ahead, you like statistics, you do something with it. So incredibly generous. And so that was Lise's, without Lise, this wouldn't have been possible. And this man here, Olivier Rey, was her technician for many, many years. And he was a gentleman that collected and did all the field work. So the same person did field work for 15 years, collecting all these ticks. And then the last person here, this is Cindy Bernard. She was still is my Swiss PhD student. She's still working on finishing her PhD. And so she was the person who analyzed these data. And we spent a lot of time analyzing the data. So the original purpose of the study that was envisioned by Lise Guerin was she wanted to do this long-term monitoring. So by the time we got to the data, there had been 15 years of monitoring of these Ixodes rucinus tick populations in Switzerland. And this data set, these sort of long-term data sets are rare and they're valuable because they allow you to look at interannual variation in the density of your arthropod population of interest. And the original purpose why Lise did this study was she wanted to see whether climate change was affecting the density of ticks and the risk of Lyme disease. But Cindy and I used the study to test this idea of whether beech masting, so the production of beech seeds by European beech trees, was important for the density of ticks and the risk of Lyme disease. So we had a single study location. That's always a trade-off, right? If you're gonna survey something for 15 years, you probably don't want to try and do it at 10 sites. So it was a single study location, which is here. We're here this is Switzerland, Western Switzerland here. This is the border with France. Geneva's down here, Zurich somewhere around here. And so we were at Neuchâtel on Lake Neuchâtel, and we are along the border here. You have the Jura Mountains, which are a very old mountain range. If you ever wondered where the word Jurassic comes from, it comes from the Jura Mountains. So most of the mountains are not much higher than 1,000 meters. So they're covered with forest. There's no high alpine zone there. And that was where our study site was located. We did have 4 different elevation sites. So we're on the same mountain. We're all on one mountain, which is called Schilman Mountain. And then we had 4 elevation sites at low, medium, high, top. And of course, there's going to be different weather here. It's going to be colder on average at the top site compared to the low site. All right. So we have these 4 elevation sites. And then Lise and Olivier established permanent sampling transects. So they always went back to the same place in the forest and sampled it year after year after year. And that's sort of shown here what that looks like. And so in case you've never sampled for ticks and you wonder what that looks like, this is what it looks like. you take a flag, preferably white, you attach it to a dowel, a stick, and then you drag it behind you. And so you drag it over the vegetation. And what that does is that these ticks, these Ixodes ticks, are sit and wait predators. So they're perched on the vegetation, waiting for a vertebrate host to pass. And so when this flag passes over top of them, they get excited, they grapple onto the flag. And you can then, once you've dragged your flag for a certain transect distance, you turn it over and you inspect it for ticks. And because the ticks are black or dark colored, they're easy to spot against the white background. I should point out that we don't return the ticks to the environment. So we put them in little tubes. We take them back to the lab to test them for tick-borne pathogens like the Lyme disease bacterium. And so this is an example of removal sampling, right? So these ticks, once we catch them and we remove them, they're not going to contribute to the population dynamics of those tick populations anymore. They can't be recaptured anymore. They're gone. So that's how we sample for ticks. And so the data that I'm going to be talking about today was based on this sampling protocol. So I introduced you to the 4 elevation sites. The study went on for at least 15 years. And every month, Olivier would go to those 4 elevation sites and he would drag that flag for 100 meters at each of the 4 elevation sites. So there were 720 sampling occasions, and on each sampling occasion, he covered an area of about 100 meters squared. And then he, counted all the ticks. And so over the 15 years, Olivier collected over 40,000 ticks. And then he brought them into the lab, and a subset of those ticks were tested for the presence of the Lyme disease pathogen using PCR-based methods. Okay. And I'll just point out that the seasonal activity of Ixodes ricinus ticks is quite complicated, and I should specify that I'm talking about the nymphs here. I forgot to say this earlier, but the nymphs are the most important stage because they're an order of magnitude more common than the adult ticks. And so really the risk of Lyme disease is dependent on the density of infected nymphs. So I forgot to say that earlier. And so what this is showing here is this is showing the seasonal phenology of nymphs within a year. And so you see it's kind of a complicated pattern. There's this early spring peak of activity, then it drops off, and then we have a second peak in the fall. And I can talk later about why that happens. So we don't have to get into the details now. What I want to say is that Cindy and we were interested in variation among years. So we weren't interested in explaining this variation. And so what we did is we simply, to know the total abundance of the ticks in the year, we simply calculated the area under the curve. Similar to what Pablo did with the, I think it was the water levels, right? So you have a complex function like that. You want to get some sort of summary statistic. A great way to do it is you calculate the area under the curve, and so you have an annual estimate of tick abundance, right? So that's what I'm talking about, and we're not interested in these seasonal variations. Okay, and then we are interested in trying to explain variation in this year-to-year variation. Sorry, we're interested in explaining year-to-year variation in tick density with the different ecological variables. So of course, we're going to include elevation site. We're going to include year. We included beach masting index with the appropriate time lag. Remember that it was a 2-year time lag. And I'm going to tell you in a little bit where we got this beach masting data from. And then we looked at climate variables, and that was fairly standard. We looked at temperature, relative humidity, saturation deficit, which is a combination of temperature and relative humidity and measures the drying power of the air and precipitation. And we would, this is a bit crude, but we would average them for the whole year. And then again, we would look at different time lags. So we would look at the mean temperature of the same year of the tick density, or the year before, or 2 years before. Okay, so we introduced time lags for our climate variables as well. And then we created linear models, and then we used an approach called AIC-based model selection, which if any of you are confronted with these kinds of problems, I encourage you to use that approach rather than sort of standard stepwise regression models. There's some real advantages that approach has. My cat is scratching at the door. I'm just going to open, otherwise he'll keep scratching. Where did we get this masting data from? We didn't get it ourselves. When we realized that masting was important and we wanted to look at it, we started looking, colleagues had told us that there's people who keep track of this. And then we came across this study in ecology, and it was this group of researchers that collected 2 centuries of masting data for the European beech and the Norway spruce across the European continent. And you were able to download this giant Excel file, and it just was for places all over Europe. It had Neuchâtel, and there it was, this data that we were looking for. So we didn't collect it ourselves. We got it from this database, and it's quite simple. They just give the intensity of masting on a 1 to 5 scale. So 1 being a very poor masting year, and 5 being a very strong masting year. So that's where we got the masting data from. And in case you're not familiar with AIC-based model selection, I just put a table up here to give you an idea what it looks like. So this table shows the top 6 models. We looked at many more models than that. We looked at more than 50. And it ranks these models based on this ACA-ICE information criterion score. So the lower the score, the better the model. It's sort of the model, the most parsimonious model that explains most of the variation in the data. So here, our top model of all the models that we looked at included the factors of site, year, the beech masting index 2 years prior, and relative humidity. And what you get, these AIC scores are then converted into weights, and that you can say that's the support or the weight that the model has. So this first model has a weight of 76%. So sorry, the weights have to add up to 100%. And the next best model has a weight of 6%. So we can say that this first model is 12 times better than the second best model. But if you see that, the top 6 models, I haven't shown all the models here, but these top 6 models, they all include the effects of site, year, and beech. So that sort of indicates that those 3 variables are clearly important. And then the 4th variable is sort of a floating variable. Sometimes it's relative humidity, sometimes it's saturation deficit. So it's important, but not as important as these other 3. So that's what model selection looks like. And a real advantage is you compare different models with multiple regression or stepwise regression methods. You always have to compare nested models, and you don't have to start with a giant model that includes all your explanatory variables. So there's some real advantages with this approach. Right. So now let's look at the relationship of these explanatory variables in our top model with tick density. So the first result was that the tick density decreased with increasing altitude, and that was not a surprise. We expected that. So tick density is high at the low elevation sites, which is about 600 meters, where it's warmer. And then at the top elevation site, that was about 1,000 meters above sea level, the tick density is really quite low. And that's probably related to those developmental times that I showed you earlier, which become very, very slow at colder temperatures. And so ticks probably don't have enough warm days to complete their development, or that's what's reducing their abundance at this top site. So this result was entirely expected and consistent with previous studies. This slide shows how the tick abundance changed over time. Initially, Lees-Guerin's expectation was that the tick abundance would increase over the course of the study, because we think that with climate change, that should generally be favorable for ticks in Switzerland. And we found increases over time at the low and medium sites. The high site was more or less stable, and at the top site, there was a very dramatic decrease. And Lees-Guerin was initially expecting that there would be a dramatic increase here. I can talk later. There's 2 explanations that we have for that I can talk about later. But so the dynamics were different at the different elevations. We found an effect of relative humidity. There was a negative effect which on tick density, that was kind of surprising because I said earlier that these ticks are very sensitive to desiccation, and that's why they live in deciduous forests. So most people that study tick ecology might be a bit surprised by this result. We don't have a very good explanation for it. Maybe too much relative humidity. Too much of any good thing is not good. In our paper that we published, we suggested that maybe we know that there's fungal pathogens that can attack ticks, and maybe they get the upper hand if it's too humid. So we don't have a very good explanation for this at the moment. But now we'll get into the main sort of part of the story, which is the beach masting. And so here we have our 4 elevation sites again, and the colored lines show how the tick density is changing over time. And then the bars refer to the beach masting intensity. And if you remember, I said that it ranges from 1 to 5, 1 being a weak year, 5 being a strong year. If you're wondering why these bars are all the same, it's because the beach masting index data that we got were only for the whole canton of Neuchâtel. They weren't, site specific. So that data is the same for all 4 elevation sites. And what I've done here, I've included that 2-year stagger. So the beach masting is actually the beach masting score 2 years before the density of nymphs was measured for that year. Now, to see the relationship here, I'm going to show you a better graph in a little bit. To see the relationship here, you would have to sort of be able to do a multiple regression in your head and simultaneously control for the effects of elevation, year, and variation in relative humidity. So that's tricky to do, but there is, you can maybe see an association that big masting years are associated with big tick years 2 years later. If you pull the parameter estimates out of your linear model, the relationship's easier to see. So this shows that there's a positive slope between the beech tree mass score, and I should have said 2 years earlier, and this cumulative nymphal density, so the tick abundance for the whole year. And to put this in a quantitative basis, if we go from a beech tree mass score of 1 to 5, our tick density 2 years later will increase by about 85%. So there's almost a doubling if the beech tree mass score goes from poor to very good. Now, as I mentioned earlier, we actually tested a subset of about 13,000 ticks for the Lyme disease pathogens. So we have estimates of the density of infected nymphs. And so if we look at that and we do the same analysis, we more or less find that it's the same. And so here again, we see a positive relationship. This time here, what we have is the DIN, that stands for the density of infected nymphs. That's a very commonly used acronym in the Lyme disease world. So we see again that there's a positive relationship between the density of infected nymphs and the beech tree mass score 2 years earlier. Okay. And so as I mentioned before, the density of infected nymphs is what really determines the risk of Lyme disease to vertebrate hosts, including humans. And again, if we go from 1 to 5, we're going to increase the density of infected nymphs by almost double. So what we can say is that in the years following a masting event where you have tons of acorns and beech nuts being produced, about 2 years later, you can see as much as a doubling in the risk of Lyme disease. So yes, I've got to my conclusion slide, so I've already said them, but I'll say it again. So we saw that the beech masting index in year T-2 increased the density of nymphs in year T. So there's a 2-year time lag. And we also saw that it had the same effect on the density of infected nymphs. And so we can say that the risk of Lyme disease will double 2 years after a big mast year. So this phenomenon had been shown before in North America, but our study is one of the first long-term studies in Europe to estimate this quantitative effect that seed production by deciduous trees has on the density of infected ticks. And so it's useful information because public health officials in Europe should be aware that the risk of Lyme disease will increased significantly 2 years after a masting event. I remember in Switzerland when I was there, there was sort of an obsession with the media every spring as to whether it would be a big tick year. And it was always, people would speculate, oh, it was a hard winter, and so we should have less ticks, or it was a mild winter, so we're going to have more ticks. But really what our long-term analysis found is what matters is what the beech trees were doing 2 years earlier. That was probably your best predictor. If you're interested in this story, we published it this year in 2020 in Parasites and Vectors, and we're actually just got our second manuscript accepted in the same journal that talks about the DIN. This one only talks about the density of uninfected nymphs before testing. And in closing, I want to thank my collaborators, Cindy Bernard, who analyzed the data, Olivier Rey, who collected all the data over 15 years, and Lise Guerin, who had the foresight and the funding to get all this sampling done. And I want to, of course, thank the funders of this research and my research at the University of Saskatchewan. And then I'm happy to take any questions about this presentation. Thank you for your attention.
Thank you so much, Maarten. So yeah, if anybody has a question, feel free to unmute yourself or use the chat box. So I do have a question. When you show the graphs of the nymphal density at the different levels and over the years, and we saw that in some cases there was an increase or maybe it was fairly constant. What's your explanation for the top level where we see that it decreases?
Okay, yeah. So there's two explanations here. One is that during the course of the study, they built an outdoor adventure park at this top site. You can take they call it a funiculaire. It's sort of like a little like a gondola that goes up the side of the mountain. You can take it to the top there, and they built an outdoor adventure park. So there's a zip line, there's a course for mountain bikes with lots of jumps, and this construction happened about 30 meters away from the top elevation site, and they didn't change the location. So one possible explanation is the construction of this site and then the increase in human visitors, and maybe, they cut down some trees and stuff. So maybe that environment was no longer very good for ticks. Maybe the vertebrate hosts were scared by all the mountain bikers. And so that's one explanation. And this is an interesting, complication with long-term field sites, right? Is that you hope that they stay constant and the same so you can change, so you can monitor changes in the environment. But if the government of Neuchâtel decides that they should build a recreation site there, your arguments that this is an important long-term field site are probably not going to stop that from happening. So that's one possible explanation. The other explanation, though, is I said earlier that when we drag with the flag, it's removal sampling, right? We remove ticks from the environment. And the working assumption is always that, you're removing some very small fraction of ticks, and nobody knows exactly how much, but people I think probably would think less than 1% or 2%. And so it doesn't have any real impact on the tick population dynamics. But maybe at this top site where the tick population is already not very big, and we go back year after year and drag every month, maybe we actually did this. Maybe our sampling did this. And it's hard to say without, appropriate controls. I mean, ideally, maybe we should have at that top site drag once a year at some other place and seeing whether so those are the two explanations: either the creation of this construction site which deteriorated the habitat, or this population of ticks is already pretty small, and then sampling it 12 times per year and removing the ticks that we did, maybe that had a negative impact. Okay. Does that answer your question?
Perfect, thank you.
Yeah.
Do we have any other questions? Oh, yeah, go, Pablo.
Yeah, very nice talk. So I think that your second explanation is much possible, because all the high, not the top, but the high site is constant, and that's because it's the population is a little larger than on the top, but it's is lower
Right.
than the median and low. So that's kind of on an effect. So that will support your second explanation. And the
that's true.
and the other one, you have to see like a very particular point when you find this drop, when this perturbance in the environment happens. So and well, here you just see one line. You don't. I don't know how the data changed.
Right.
Particulars.
Yeah, no, I agree, and I'm partial to this explanation. I came up with the preferred explanation a little bit later. At first, we and I should say that the construction of the adventure park, it went on, and it wasn't like it was done in 1 year. Like they first decided to add this, and then they added that, and then they added this. So it was done sort of over. But I agree that you know this explanation that would fit nicely with the eyesight. Yeah.
And it's not possible to estimate the total population on each site, or by some kind of, I don't know, some estimation of the numbers.
Well, that's always the difficult thing, right? I mean, it's often with our sampling methods, you have an index, but you don't know exactly the true population size. I mean, that's it's for some species, you know, we know that there's about 500 whooping cranes in North America, but for many things, you have sort of a rough idea maybe. But I mean, as I said, like, I don't know, are we sampling 1% of the tick population, 5%, 10%? You know, so it's hard for us to answer that question, but yeah, so it's just really more of a relative index, right? Is how I look at it. It gives you an indication of the abundance, but to know the absolute amount of ticks in the environment is not easy to calculate. You'd have to do some other methods to really get at that.
And one last question, because you have another question on the chat. So this regression with the relative humidity, if you do the same with the water vapor saturation deficit, is the same relation, or it changed? Because there, as you say, you get into, you also use the temperature, but
yeah,
here is only the humidity.
Yeah. I have to remember now how I think was set. So I think temperature had a positive effect, and then saturation deficit, because of the way it combines temperature, saturation deficit also had a negative effect. But that's the nice thing about this AIC-based model selection. If we go back here, you know, here we have the first top model is relative humidity, and then saturation deficits down here. So you would think, you know, at first principles, you would think saturation deficit should be better. But it has 5% of the support, and this one has 76% of the support. So
yeah, it's true.
So, but I just want to acknowledge a real limitation of, well, I mean, I think the beach masting story is, you know, I'm very confident in that, and I think that's the exciting thing about what I talked about today. I think what we realized when we did this modeling is we were very restricted or we weren't very imaginative in how we thought about climate, right? Because when you think about it, you can have a year that has a very, say, hot summer and then a cold winter. And if you calculate the average temperature, it'll be some value. And now imagine you have a year, we have a mediocre winter and a mediocre summer. And that can have the exact same value as the previous year. How the ticks will experience that year, a cold winter and a hot, you know, and a hot summer, is very different. But when you take a mean, this is obviously really crude, right? And actually this gets to, I've found this very interesting in trying to think about this work. What is the relative time span over which we should be looking at these climate variables, right? Is it a whole year? Is it a month? Is it the winter? And so in the modeling that I'm now doing with Cindy, we're actually on our third pass on this data set. And we're now trying to explain more of the seasonal dynamics that I showed earlier. And now what we're really trying to do is, you know, you take the year and then you chop it up into seasons, and then you have the time lags. But the problem is that you can easily construct, dozens, if not hundreds of different climate variables, right? That are over different durations and different time lags, and so on. So it becomes, and at the end of the day, even though this is a lot of work, this is not an enormous data set. There's only 15 years here at 4 elevations. So we have 60 data points if you look at it at an annual basis. So that's a really interesting challenge, is knowing, what are the relevant time spans that we should be averaging these climate variables over. So, and we hope to get a better understanding of that in the future. Yeah.
Thank you, Maarten.
Yeah, you're welcome. And I don't see the chat, so maybe someone can.
I can read that for you. So this is another question from Matt Unick, and he says, thank you for the talk. Would rodent infestation intensity increase in years when I assume it's would tick infestation density increase in years when rodent populations are lower? And if so, would that impact the DIN also? Was there much variation in the understory between elevations?
Okay, so I'll answer the first question first, and it's a very good question. And I spent a lot of time, Cindy and I spent a lot of time thinking about this, because it's actually quite counterintuitive. Because what happens is, what does Lyme disease transmission depend on? Lyme disease transmission depends on nymphs and larvae that are actually different cohorts, right? But active in the same year. They have to feed on the same host. So a good thought experiment is to just imagine that there's only one mouse in the forest. Make it a very big mouse, make it the size of a house. But if it feeds all the ticks, that would be the best from the pathogens perspective, right? Because all the nymphs will get on there and inject it with Lyme disease spirochetes, and all the larvae will feed on that mouse. So what happens if the rodent population explodes? Well, then you get fewer ticks per mouse, right? And if you make the experiment, make an infinite number of mice, each tick will have its own mouse. And if that happens, there would be no transmission. And actually, this is a very general prediction of models of vector-borne diseases, which is that the force of transmission depends on the ratio of the arthropod vectors to the hosts. And so once a massing event explodes the rodent population, even though there's lots of food for the ticks, which is good, there's actually kind of a dilution effect, which is that if there's not aggregated feeding, if nymphs and ticks are not feeding on the same hosts, then the transmission can go down. So you have two different things that are going on. Transmission could actually be slightly lower in one of these in the years after massing event, because rodent density is so high. But because you have so many more larval ticks being recruited into the nymphal population the following year, that makes up for it. So it's a very good question. But so high rodent density can actually dilute the transmission, but because you have so many more larvae getting a blood meal, it makes up for it. But they sort of work in opposition to each other. And at first we thought that the percentage of infected ticks should increase following a mast year. But when we thought about it more, that's not necessarily the case. So that was your first question. And your second question was about whether the vegetation differs. And that's a very good point. And so, of course, it does. I mean, there are some changes. I mean, I said it was forested, but there are changes in the vegetation. And so one possible explanation too that we can't, it's difficult to answer here is our dragging efficiency the same at all of these habitats? If it's known that if your vegetation structure is different, that's going to influence your dragging success. So that's a factor to consider. The vegetation is not the same at these 4 elevation sites. But again, we're very confident in the results of the beach masting, just because it was I forgot to say, so beach masting explained about 25% of the variation in the density of ticks with that 2-year time lag. So, for those of you that do long-term ecological studies, you know that it's often not easy to explain a lot of variation. But so we found that 25% of the variation in interannual variation of tick density was explained by beach masting. And I didn't say this, but we looked at different time lags. So we looked at 1 year time lag, 2 year time lag, 0 year time lag, 3, 4, 5. And it was only the 2 year time lag that had this very strong effect. So 1 year time lag, not significant. 3 year time lag, not significant. Only 2 years time lag was important. So that makes us very confident about the beach masting story. Thank you.
Do we have any other questions?
Oh, great. Thank you.
Okay, thank you so much. And I guess that one last question. Do you think you will get to repeat something like this in some other place in Switzerland?
Like at another site to test it again?
Exactly.
Well, I think what's really interesting is how difficult it is to get funding for long-term studies like this, right? And there was recently that study I think it was published in PLOS One, where this German entomological society showed that insect abundance had decreased 80%, right? And I think it was one of the most tweeted and talked about publications in 2018 or whenever it came out. And it was done over 80 years, but it was done by an amateur entomological society in Germany, right? It was funded by the government. And that's a real shame, is I feel like we need money for long-term studies, but you ask the government, say, I want to do a 15, 20-year monitoring study. What do you think? I mean, people are going to run away. So it's hard to do. And, we're lucky that there are, like the beach masting data set that was on the internet. And, there's some things like that people have. And there's some countries that are keeping good records of Lyme disease. So there've also been people that have correlated masting. There's studies that have correlated masting with incidence of Lyme disease. There's one study that came out in Poland that looked at that, again, with the 2 year time lag. So the incidence of Lyme disease in human population goes up because those two things are being monitored. But it's hard to get money for this kind of work where you say, I want to go sample abundance of ticks or insects, arthropods for 15 years. And Leesgern got it, but got this money. But I don't think these studies are very, very valuable, but there's not a lot of interest by governments to fund this kind of research, unfortunately.
Okay, thank you so much, Maarten.
Yeah.
So if those are all the questions, I guess we are done for today.